Hi, everyone, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today we are talking to Marco Pagani from the Functional Imaging Laboratory of Istituto Italiano di Tecnologia in Italy and the Autism Center of the Child Mind Institute in New York. Marco uses magnetic resonance imaging to study the functional architecture of the brain in both humans and rodents and the changes that occur in pathological states. We're going to be talking to him mainly about autism today, I think, although who knows where it will go exactly. Welcome, Marco, and thanks for joining us.
Hi, and thank you for having me here today.
With us today we have Alice Bertero, who's a local expert on cortical connectivity. Hi, Alice.
Hello.
And Nicole Wicha, our expert on cognition, language, and the brain. Hi, Nicole.
Hi.
And I'm your host, Charlie Wilson. So, Marco, there's a pressing need to understand the causes of autism and to find treatments for it. And autism isn't just one thing, with just one cause. And so we need to be looking for more than one treatment, probably. You can tell me when I'm wrong.
Mm-hmm.
So
I completely agree with you, of course.
There's several different ways to try to categorize it. We could categorize it by behavioral phenotype. I'm sure there are people who do that. Or by genetic etiology and by measures of brain function. There might be other ways. But the first question is whether all of these form clusters or they're just a continuous distribution of anything that we measure like that. And if they do form clusters, can we clarify things by creating categories? And then if we can create categories, which I hope we can, then do those categories map onto each other so we could find relationships between behavior, genetics, and brain measures, like the ones you measure using functional MRI?
Mm-hmm. Yeah, thanks. These are, of course, very interesting question. And what I can say is that for like decades, especially neuropsychiatrists have used techniques like EEG or functional MRI or other neuroimaging techniques to kind of correlate what they see during their clinical practice in the behaviors of children with autism, and try to kind of link those behaviors to brain systems. Okay, so this was like the big challenge, I think, of the 90s over like early 2000s. And of course, that approach has been extremely successful because it has enabled us to characterize, to link a little bit these two different scales of measurement, so behavior and brain circuits. However, of course, there are some. It has now become evident that there are lots of limitations in this approach. And one of these limitations is exactly what you said, that basically all these measurements relied on the idea that there is just one single form of autism. And then, what happened is that basically we had lots of neuroimaging studies applying measurements such as, for example, functional connectivity, which is this measurement that can allow to map how different brain region interacts, and people have applied this technique to better understand autism. However, what was the results of all these studies? Was that it was a lot of heterogeneity, like you said. So basically, what they were doing is basically mapping in each single study some subtype of autism, and they couldn't replicate the findings across studies or across population. And so, one idea that we had, but also other people, other laboratories have, are started to develop, is that, there is no, that all this heterogeneity was not just simply because what we are mapping is like our technique are noisy, or just because the methods that we use are not reproducible, but instead because we are mapping some heterogeneity that can have neurobiological importance, relevance. And one idea that I think we developed during the time is that most of this variability, autism, so it has a lot of variability in terms of behavior. So if you talk with a child psychiatrist or a child psychologist, what they tell you is that basically each patient is different. Children with autism is different from any other. Right, so there is like this landscape, this very spectrum of variability, and also neuroimaging studies have kind of identified this variability at the brain system level. But also, what we know so very well, what we learned out of genetic studies is that there is also lots of variability in terms of genetics. So because there is no one cause, as you said, exactly, there is no one cause for autism, but there are multiple. Okay, we know what we know very well, and there are some of this variability can be explained also by environmental factors, like maybe neurodevelopmental trajectories. However, most of this variability in the causes of autism is because there are so many genes involved in the disorder, right? So there are so many. And then, what we're trying to do with our research is kind of find to use machine learning approaches, statistical, mathematical approaches to find these subtypes of autism based on by using fMRI. Why fMRI? Because autism is a brain disorder, so perhaps the secret to kind of dissect the different brain circuits is like to use techniques that are sensitive to brain activity. And what we found so far is that yes, we can use brain imaging measurements, like for example fMRI, to identify clusters. So there are clusters, so there is no such a thing like a single signature of autism in the brain, but instead there are many, and these many can but it's not like the case. So some people think that including us, think that the autism is a spectrum. Okay, so it's like it's a sort of gradient of continuous variability, right, across subjects. But of course, if you want to kind of simplify a little bit and make inference out of this data, what you can do is to identify these clusters and put together patients that have similar connectivity alterations. Okay. And this is basically the approach that has been used so far by clinical neuroimagers, and has been extremely successful because they show that there are some subgroups of autism. There is no one signature. There are multiple autism, and in fact, lots of people stop to call it autism, but they call it autisms because there is multiple. There are multiple forms. However, this approach, is extremely powerful, and it just brought us to a complete change of perspective in the way we study, we understand, we approach the complexity of autism. However, one missing link here would be that, of course, it's very hard to study genetics in the humans, especially to have, both data that you can use for your measurements with genetics and brain imaging, because there are so many different forms of autism that these etiologies are extremely rare. So it's very hard to find, to study, for example, the brain circuits of specific genetically determined population of autism. And so what we, our idea, our way, which is to kind of address all these questions would be is like to use animal models. So we use the mouse. We use the mouse because the mouse is we call it like the quintessential tool to study the genetics of autism, because there's lots of different models that are available for autism. And then what we do is to use fMRI to map the brain circuits that are altered and are associated to that specific genetic etiology. And of course, that adds a lot. Of course, it's not an approach that is in contrast to what people do in the clinics, in the clinical setting. It's just a way that, it's just measurements that are aimed to complement these measurements and try to go a little bit deeper to investigate the genetics.
When thinking about clustering, and we're talking about finding clusters in some space, so in the genetic space. When thinking about clustering, we think about a space that has a bunch of dimensions in it, and now we're looking for clusters in this high-dimensional space. So I'm kind of interested in how many dimensions there are. So there are many different genes that influence autism. Are there single gene versions of autism? So there are genes you could say, if you have a mutation in this gene, you do have autism. Mm-hmm. Or is it one of these things where you have a 50% risk if you have this?
Yeah, I see. Yeah, thanks a lot for this question. Yeah, I think it's more like the second that you said. So basically, there is no such a thing like a gene that if it's mutated is like is you have like 100% of chances to get a diagnosis of autism. But we kind of say that these genes that are identified to be related, to be linked to autism, they're like risk genes. So basically, if you have a mutation in that gene, you are more likely to get a diagnosis of autism. So it's not like one-to-one correspondence.
To make a mouse model of autism, right?
Mm-hmm. I was going to ask that because there's something that's been very curious to me about when designing mouse models, when we don't actually know what the genes are. Some people have approached this by trying to model the behavior. Mm-hmm. In your case, it doesn't look like you're modeling the behavior. You're picking a gene that does what? That does what? Like, what are you mapping the assumption of that this is a model of autism based on your...
Yeah, I see. So first of all, sometimes scientists, including me, we refer to these mice as like mouse models of autism. Of course, autism is something that is for humans, so there is no autism in the mouse. However, what you can do, you can use genetic engineering to build these mice like a mouse that have that carries the same mutations in the gene that has been associated to autism in humans. So this is what you can do so you're not like studying with the mouse, you're not studying autism, but you're studying what that specific mutation does in terms of brain circuits or whatever you're interested. And you kind of use these measurements to cross compare with the clinics.
So you don't require the mouse to actually have autism.
Yeah, so behavioral symptoms. But you can measure then behaviors also in the mouse. Of course, a mouse they're like and actually they are very good models also for behaviors because like mice are very social. So, and of course, social behaviors is what is one of the core impairments in clinical autism. So, like the mouse model is absolutely a great model to study also autism behaviorally. Of course, the repertoire of mice behavior is much more narrow as compared to of the social behaviors that humans can have. But I think it's still a very valuable model.
So I think that the methodologies that you're using, like linking the functional brain activity networks to the specific genes, especially they're doing that co-correlation mapping of the genes that are present. So you're actually starting with a gene that's been suggested in the human literature, looking at it in the animal, and then correlating it back to functional neural networks of some sort.
And behavior.
And behavior, but it's just one gene. And so how do you I mean, it's going to be really frustrating as someone who understands the complexity of autism to have one model of maybe something that has something to do with autism, right? Like, it's connected in some way, but when you end up with your network of brain regions that is involved and you're saying this is the network that is deficient in autism, how do you feel as a researcher talking about that and your ability to sort of generalize that to other aspects of the spectrum?
Mm-hmm.
And like, how much is that really autism, or just that particular behavior or that particular gene?
Yeah, this is an amazing question. So first of all, I would like to get a step back on your question that you asked before. So how do we select these genes, and so why exactly that gene and not another one? And this is that was also an incredible question. Thank you for asking. And basically, so that comes from the human literature, and usually in human literature they have these very large scale genetical study. They call it like GWAS, and so that's a way that people basically use. They have these very large populations, and they do like genetical screening, and they can kind of filter out some genes that are associated to autism. And also the other way is that they're in human genetics. People use family studies. Okay, so basically the people follow scientists follow these families where there is at least one case of autism, and they kind of see how that mutation is present, for example, in the children with autism, and if that was not present in the genitors, so that would point at the possible link of that gene with the mutation in that gene with autism. So what we do is basically just to generate a mouse model with that one. So when we have a strong candidate, and there are also foundations that of course are generate a curated data set, so you can consult this data set and get to know whether that gene has been in the literature, in the scientific literature, has been highly involved, highly linked. So at which degree that gene was linked with autism based on these large scale studies.
So you have an advantage, I guess, because there's a huge number of mouse models that have been generated using this genetic approach.
Mm-hmm.
Like I looked it up. There's 1,353 of them. And
that's lots of work for us.
So, but it does mean that you get to take your pick. You don't have to pick the ones that are most weakly connected to autism. You can pick the ones that you think have the most strong connection.
Mm-hmm.
So is that what you do? You search through them and find one that isn't a mouse that isn't like 50% likely to be autistic, but is more like 90%.
Yeah, so
and also you choose your models also based on the phenotype of the mouse model that's been generated. So let's say that this mutation has a 50% incidence in this population and is strongly correlated with humans. Then the mouse has been generated and characterized, because that's the first thing that you do when you generate a model. And if this mouse shows some behavioral, or usually the behavior is the first thing, or some gross abnormalities that are correlated, would you consider this more than another one that shows less or weakest behavioral phenotype?
So yes, of course.
Thanks. All categories, right?
For your work, you can't be studying thousands of different mouses.
No, it's
sooner or later.
I will need 10 lives. There's so much time to characterize them all, and also, there are the number that you said is already overwhelming. So there are so many genes that could be involved in autism, and we're counting. So basically, every time I search the scientific literature, or just I'm very active on Twitter. Every time I wake up and I have my cup of coffee, there is a new gene that has been involved in autism. So there was a certain point when I started in Italy, the Italian Institute of Technology with Alessandro Gozzi, which is my was the senior scientist I was working with in Italy. We basically knew all these genes by name, because they were and it was only 10 years ago. Now there are so many that it would be impossible to remember even only the name for all of them, right? So so how to prioritize which gene to investigate? That's an amazing question. Of course, everything you said, it's something that we factor in. So behavior, and so usually what we also take into account is that, so we really want, so our one of our main interests is that is to find some correlate with MRI or with fMRI. So basically, one of our things that we're investing the most is to use fMRI and to use MRI on these mouse models, because we really want to use a technique that is translational across species. So in the sense that, of course, MRI and fMRI is exactly the same one that it can be used in the humans. And what we are very interested in is to see whether we can kind of reconstitute the same abnormality, the same atypicality that in the mouse model, the same that we see in the humans. But then the next step, if we are able to show that, then the next step is try to go, not myself because I'm not a molecular biologist, but I work with a lot of biologists, and of course then the next question is, well, okay, great, so we were able to reconstitute the same MRI phenotype, the same MRI alteration, but then now what's next? So one point is that we want to use this mouse because to use all the beautiful neuroscience techniques that you can use on the mouse, and deeply characterize for example, what happens at the microcircuits, what happens at the cellular level. So these are all questions that we are also interested in. So like when we kind of try to prioritize one gene instead of another is because perhaps that specific mouse model has been identified a specific cellular signature, maybe related to number of neurons or number of synapses, or like all these basic cellular features, so that we can kind of trying to link our functional measurements at the macro scale that we can do with fMRI, and these all the measurements that are made at the cellular level. So this is also having also a deep biological characterization of that specific gene in the specific mouse model. That's also something that we also factor in together, of course, that with the behavior, which is always one of the first things that you want to know about that mouse model.
So the genes cluster in sort of functional space of their own. So some genes are all related to each other. They're part of some pathway, for example. And so you could say, well, these genes ought to be similar in effect. If the mutation in any of them is like messing up this one pathway, then all of them will be approximately the same kind of thing. So if you collapse the enormous number of genes down along functional lines like that, you end up with a lot smaller number of pathways that you're thinking about. And one of them that you focus on is the mTOR pathway. And I guess not just you, but that's been a focus of lots of people who are working on autism. And one of the attractions of that is the observation of this cellular change, which is an overabundance of dendritic spines. So that's the kind of clue that you're looking for. How common is that? Is that just for these mouse lines with the mTOR pathway mutations, or is that is that something that's seen across many different cellular pathway?
Yeah. So for now, basically, I think that lots of the inspiration of the research that we do at the IIT starts also from clinical data. Okay, so basically what we're trying to do, with all our work with the mouse models is like, okay, we use the mouse as the tool, but of course the question is always clinical. So what we are trying to do is like to make our research as translatable as possible, in order also to speed up the piece of discovery in the humans. So for now, we focused mainly on not mainly, but at least for my research, I focused a lot on the mTOR pathway, because the mTOR pathway has been found to be disrupted, actually hyperactive, in in postmortem brains of children with autism, and so that's why we were interested also to study a mouse model with the same mTOR hyperactivity. And as you said, like the mTOR pathway is extremely complex. So if you look at the textbooks, there are like so many genes, so many interactions involved, and among all of them, we used we investigated a specific mouse model. And this specific mouse model is the TSC2. And why the TSC2? Because this TSC2 mouse model has been shown to clearly reconstitute this synaptic surplus that it was exactly the same that was found in the human literature, right? So we have always this parallel between the species, and in fact, this is like, I think we've been one of the first lab who invested a lot in doing cross-species study. So when most of the people think that fMRI is something, or MRI in general can be done only in humans, instead we say no. Listen, we can leverage the power of these cross-species studies, and use it and couple it with the power that mouse models can give to really advance our understanding on the origin and significance of the functional connectivity alterations in autism.
So dendritic spines on cortical pyramidal cells, that's the observation, right?
Mm-hmm.
And the idea is that every dendritic spine gets an excitatory synapse. I guess that's definitely been shown under sort of normal circumstances, and I think it's probably reasonable to assume that that's true in this case too. And so that means if there's more spines, there's more excitatory synapses on cortical pyramidal cells. So there should be more excitation in cortical pyramidal cells, and that somehow that could be linked to changes in the connectivity that you see with your... So I'm just sort of trying to see how this all fits together.
Yeah. So yes, that's for sure. And in fact, another common observations that we have on these mouse models is that these mouse models are epileptic. So they do have epilepsy, and this, of course, is related to the higher excitability that you mentioned. So yes.
And also synchronization, right? Which is what you're measuring in your data.
Exactly, and also
hypersynchronization.
And also hypersynchronization, hypercoupling across brain regions. So that's so both number of synapses, but also the fact that these synapses are excitatory. Of course, it's could be are two factors crucial that can crucially explain all the hyperconnectivity that we measure. At the network level. On the other side, I think that until a few years ago, people invested a lot, and people are still investing a lot in understanding whether the importance of, for example, measurements of E2I imbalance. And disruptions in E2I balance can lead to some disruptions in networks in people with ASD. So nowadays there are like more and more studies that, however, are kind of like putting all these findings in a different light, in the sense that people now, and there are like many publications that are, and especially a couple very influential, that instead shows that these E2I imbalances are not like a cause. So this is not like the initial cause of autism, but instead are like a ring in the chain. So basically something in the mid,
maybe maybe an effect rather than
maybe an effect instead of a cause.
I should say something about what E2I balance means, right? Excitatory and inhibitory.
Sorry, excitatory and inhibitory.
And so that means that you could have more excitation. If you had more inhibition, you'd end up in kind of the same place that you started. So we wouldn't necessarily have to have more activity every time we get more excitatory synapses. So how do you evaluate that? I mean, what's the test for that? I know that's really not something that comes straight out of MRI.
No,
very easily.
No, that's not. But there are like lots of studies that now are, there is like lots of interest because now there are like available lots of manipulations that can enable to change these excitatory and inhibitory ratio. There are, for example, there are like sort of manipulations that allows you to excite brain regions or inhibit the activity of brain regions. And of course, what is very interesting that you do with fMRI is to map what are the effects of these imbalances in terms of at the large scale level, at the level of fMRI.
We could do something like suppress inhibition in some cortical area or enhance excitation in some cortical area, and then see what that does to the resting state connectivity.
Exactly.
It can be global with drugs, it can be local with specific manipulation. Yeah.
Can you do optogenetics and fMRI?
You can do opto-fMRI, you can do chemo-fMRI, so you can couple chemogenetics with fMRI. And there are already in the literature beautiful examples. And so also with, by using drugs.
Yeah, that was the starting point. I first blanket with drugs so that can kind of act on one population or another. And now the field is more going through opto fMRI and chemo fMRI to be more specific on cell type. That's also a beauty of the mouse model, that you can select one cell type across others.
Do we know something because of that? Or could we draw some kind of conclusion about the work that's been done with that, or is that just getting started?
So I think it's moving. It's a field that is moving very fast, and so results. There is already some very results. There are not like convergences yet, but what I can say that there are also some unexpected results that people would not hypothesize this way, but it turned out to be like these. And again, I don't want to do like self-promotion of the lab of the Italian Institute of Technology where I come, but it has been published very, very recently, this paper by a PhD student of the lab, and saying that they were switching off entirely the prefrontal cortex of the mouse brain. And of course, what most of the people would expect after switching off a brain region is people would expect that connectivity would decrease, right? Because there is like less activity, so less connectivity. Instead, what we found is exactly the opposite, right? And what happened, what we found is that when you switch off like these very crucial integrative brain regions, like the prefrontal cortex, you instead get higher connectivity, functional connectivity. And I think that this is very, these results could be extremely important, especially in the interpretation of the results, because most of the times, especially we are prone to see, so when you, especially in autism, when you see there is like functional hyperconnectivity in some subpopulation of autism, you are tempted to conclude that there is like more activity, right? And so more connectivity. Instead could be also like all the way around, okay? So, and I think that here, and of course this was modulation on all neurons, but of course, if we really want to know better the story and have a more full picture, we perhaps have to go with more targeted cell type studies with cell type specificity. So that's one way to go to better dissect these.
We're going to end up not just learning something about autism, but learning something about what we're really measuring in these functional connectivity studies.
Yeah, in the origin of this phenomenon.
Well, thanks a lot, Marco.
Thank you very much for having me here.
Nicole. This has been Neuroscientists Talk Shop.